Let's hear it.
Hi, my name is Jinling. I am a third-year PhD candidate at Case Western in the Department of System Biology. I did my undergraduate study major in biomedical sciences at Durham University in the UK. And then I went to Johns Hopkins at Dr. Connor McMeeman Lab in the Department of Molecular Microbiology and Immunology, and my master's study was using tissue-specific CRISPR-Cas9 to knock out a gene called GLOB2 and how that knockout affect the carbon dioxide sensing system in the yellow fever mosquito Aedes aegypti. And I have been joining the lab for 2 years, and today I'm going to talk about my PhD work that I have been working for over a year. My study focused on KRAS G12C mutation, one of the most common oncogenic drivers in non-small cell lung cancer, and understand the mechanisms of sotorasib resistance, a FDA-approved drug to treat this disease. And with the aim of developing strategies to delay resistance and extend the treatment efficacy. To do that, we built a platform by repeatedly expose a cell line H358 to sotorasib using multiple independent replicates across 8 evolutionary cycles that last approximately over 3 months. And at each cycles, we collect multiple types of data, including the collateral screening that to a panel of FDA-approved drugs to identify pharmacological vulnerabilities evolved through time. And right now we're doing the bark RNA sequencing analysis, and we try to answer how the transcriptional heterogeneity evolves across cycles. And these principal component analysis plot shows all the controls replicates clustered together, and while sample exposed to sotorasib shoot away in transcriptomic in space, and by the end, the directionality of the evolution may be divergent, and this complex schema reinforces these patterns. And also to move forward and connect these transcriptional shifts to pathway-specific and cancer-focused processes, we applied gene set enrichment analysis, and we identified the significant upregulation of mTOR signaling pathway consistent with the potential reactivation of downstream RAS signaling pathway to escape the inhibition. And also we could use like the heat map to dig into these significant genes. And another question we are asking, and also mentioned by other folks in the lab, is to use the GAM assay, and I also apply that framework using my experimental model, and to answer how does the growth rate change with the presence of the other species and also at different conditions. And here is actually the preliminary data from my project, and demonstrated that the competition outcomes actually shifts when we across different sotorasib concentrations. And by these two figures, you could see like when you increase the proportions of the resistant cells, which colored by red, the growth rates of the sensitive cells actually increases, which can like is a scenario of like one growth of population support the growth of another population. So overall, this project, we provide novel insight into both the ecological and evolutionary mechanisms underlying the sotorasib resistance.
How many controls?
So we have 2 independent replicates in this control group that exposed to DMSO along the time, and also 6 independent replicates in this resistant group. And throughout like the experimental process, each independent replicates was passaged into the next one separately to preserve its evolutionary history.